तुम एक काम करो, तुम थोड़ा पीछे जाके बैठो, तुम्हारे चक्कर से और जब मूव करो तो इस तरह पानी खींचेगा ना, मैं जाऊंगी और शरद कपड़ा लेके रखने के लिए these are the boys जिनको मम्मी लोग घर पे बिल्कुल काम नहीं कराते। हाँ देखो, his mother and asking to give enough work at home so that he learns how to manage himself. Okay, what did we learn the other day? We were talking about structure of chromosomes, ho gaya almost, right? We are almost done with that chapter, the first part of the chapter on structure of chromosomes, and we have to look into the next part of the chapter today. Do you all have your pencils? Kina? Okay. So, we had already studied the basic concept of structure of chromosome, DNA. We had already studied the concept of it. Structure of chromosome, we had already studied. We had already studied structure of chromosome, we had already studied DNA, types of chromosome, the parts of chromosome rather. You go back and sit. तुमको भी जाना है उसके साथ, बेस्ट फ्रेंड्स हो, पानी तुम्हारे तरफ नहीं आएगा, मेरे तरफ आएगा, and by chance I start walking over the water, my bones are surely going to crack. Now, when we talk about the concept of chromosome. So this part already we are done. First of all, I'll move into the concept of cell division today. Cell cycle थोड़ा बाद में I will take. Cell cycle I'm not going to take now. Cell cycle we will not understand the continuity. We will not understand now. If I talk about, पहले हम concept of cell division सीखेंगे, then we learn about cell cycle. Now, cell division, a basic concept of cell division introduction already we have done in the previous lectures. Off and on, I have been always talking about mitosis and meiosis, but now in detail we have to study the concept of mitosis first of all. Cell division. Now, what is the meaning of cell division? Cell division, why is it necessary? For the process of growth, for the process of repair, for the process of maintenance, cell division is a must. Yes, सबसे important. Why? For the process of growth. Cell division is very much necessary for the process of growth. Okay, now if I talk about what are the types of cell division and what types of cell division happen in what type of cells of our body, then in that case, to talk about the introduction about the concepts of cell division, let us see here. Types of cell division. Types of cell division. If that is what I am teaching you, then I have to write that there are two types of cell division. एक हो गया mitosis. सर ने आज बहुत मैच कराया क्या? Mitosis and दूसरा है meiosis. Two types of cell division. Mitosis, meiosis. Now basic differences पहले हम सीखेंगे. Then we enter into the details of this. This little bit only you have to learn according to your syllabus. But हाँ, some stages differences तुम्हें समझ में आना चाहिए. Let us see this first. Mitosis. What is mitosis? A type of cell division where this is what I am showing you. A type of cell division where this is the parent cell getting divided into two daughter cells. This is the most common type of division which is happening in our body. This is the most common type of division which is happening in the body of a plant. This is the most common type of division happening in all living organisms. And this is the method of division also which is happening in single cell organisms as a mode of reproduction. Some examples like, if I talk about how does the amoeba reproduce, by this method almost. But no, for us it is not only the sole method for reproduction. For us, it is a method where growth is also, or it is a method which also involves the process of growth, which is necessary for the process of growth. It is a method which is necessary for the process of repair. It is a method which is also necessary for the process of reproduction. So mitosis, and if I show you a concept, I don't know, let me check if I have right now any. Okay, meiosis, right now let me just tell you. If I have to talk about the concept of meiosis, then you want to understand that meiosis means apart from two cells. When I say mitosis means the process of cell division in which a parent cell divides to form two cells. If I have to talk about meiosis, the parent cell will divide to form four cells. Okay. So let us try understanding some differences between mitosis and meiosis. Some differences between mitosis and meiosis. What is it? If I have to talk about mitosis, from one parent cell, how many daughter cells we get? Two daughter cells. From one parent cell, two daughter cells. Meiosis, from one parent cell, we are supposed to get four daughter cells. Four daughter cells. Something more, we try understanding that if it is a method of mitosis, then what type of cells are involved in this process? Which are the cells of our body which is undergoing mitosis? I may understand the liver cells of our body undergo mitosis. I understand that the bone cells undergo mitosis, stomach cells undergo mitosis, muscle cells undergo mitosis. But exceptions are the cells of the reproductive organs which does not only undergo mitosis, it undergoes meiosis also. But are the cells of the skin undergoing meiosis? No, never. Are the cells of the stomach, intestine, the muscles and the liver undergoing meiosis? Never. So these group of cells which are the non-reproductive cells of our body are called somatic cells. तो जब भी माइटोसिस है तो माइटोसिस is always associated with somatic cells. Please remember the differences that I am speaking now. First of all, what I said, एक तो ये है सबसे important difference हमेशा याद रखना. Now we talk about the type of cells involved. If it is mitosis, type of cells where process happens, somatic. What is the definition of somatic? Non-reproductive cells. Non-reproductive. And this side what will be right? Germinal cells. Germinal cells में meiosis होता है. Germinal. What is the meaning of germinal cells? Reproductive cells. Reproductive cells में meiosis होता है. एक और जो बहुत important difference we have to always remember is what about the chromosome number? Abhi, this is a cell. Inside the nucleus there are 4 chromosomes. You know the concept of chromosomes now. Inside the nucleus there are 4 chromosomes. Abhi, if it is a concept of mitosis, the cell divides into 2 daughter cells and both the daughter cells have got 4 chromosomes each. Whatever the method we learn later. Both the daughter cells have got 4 chromosomes each. So what is this concept known as? Chromosome number is maintained. What is the meaning of this? Maintained matlab same in the parent as well as in the daughter cell. Chromosome number maintained. Don't be busy writing anything. जो जरूरी है I will tell you. समझो फिर. Chromosome number maintained. मतलब क्या? Parent cell and daughter cells have got the same number of chromosomes. But is it not the same here? No, here the method is different. I talk about a parent cell which has got 4 chromosomes. First stage of division. Now this particular division is more prolonged. This stage of division is more lengthier. A long phase of division, I should say a complex phase of division. This is simpler. This is a complex phase of division which has got 2 basic stages. Phase 1, phase 2. इसमें ऐसा कुछ नहीं, just one simple. So यहाँ पे in the first phase, what happens? The cell, parent cell, divides to form 2 cells. And the number of chromosomes remains same. First phase में. अब second phase में क्या हो रहा है? The division is still not complete. Second phase में क्या हो रहा है? This cell is again getting divided into 2 more. This cell is again getting divided into 2 more. And this cell is getting divided into 2 more. Number of chromosomes is half. This cell is getting divided into 2 more. Number of chromosomes is getting half. So here we will say chromosome number is reduced to half. Chromosome number is reduced to half. So अगर ये rule है, then if I talk about a cell which has got 16 chromosomes in the parental cell, तो daughter cells में chromosome number कितनी होनी चाहिए? 8 each. 8, 8, 8, 8. And number of cells कितना होना चाहिए? 4. So basic concept पहले ये समझो कि if I am talking about the process of mitosis, parent cell will give rise to 2 daughter cells. If I am talking about meiosis, parent cell will give rise to 4 daughter cells. If I am talking about meiosis, chromosome number जो भी हो, whatever chromosome number you talk about, a plant or an animal, whatever chromosome number you talk about, it will be reduced to half. 4 है 2, 16 है 8, 20 है 10. Is it possible? 7. 7 is possible. Generally not. Chromosomes are always paired. Paired. So now, whenever we are talking about this concept, तो पहले तो ये याद रखो कि chromosomes whenever we understand, chromosomes when we understand that yes, somatic cells, germinal cells, a major category of cells that we have in our body, because our body can be divided into reproductive part and non-reproductive part. Now, apart from this, what is common? What is common are some of the primary phases. Now let us try to understand the phases of the process of cell division, the phases of the process of cell division. So when we are talking about the phases, what does this mean? How does a cell progress? Meaning, I'm talking about, now say suppose I talk about like this. I am talking about a parent cell and this parent cell is dividing to form two young cells. These are the new cells. What are we learning? We are learning how does this young cell proceed to again become mature and ready to divide. This is what we are learning. We are learning when a new cell is getting formed, what are the stages that new cell go through? What are the changes the new cell go through? This is what is known as phases or stages of cell division. तो इसके अंदर actually हम क्या सीखेंगे? We are going to learn how does this small young cell start its process, its journey, just like human, no? We talk about the infant stages. 1 month, 2 months, and then progressively so many changes. And today, if you talk about yourself, it's so strange, no? That you have, or we all have, we all took birth as a small infant, no teeth, no hair, nothing, and then we are well flourished individuals today. So exactly same way, we talk about a young cell, and internally so many changes happen. And then the cell is again ready for the process of division, and again it undergoes the process of growth, and again it undergoes the process of division. So let us get started to understand what are some of the stages. The stages that we talk about is first interphase. Get acquainted with the names interphase. Then we talk about prophase. Then we talk about metaphase. Then we talk about anaphase, and then we talk about telophase. We talk about telophase, and then we talk about cytokinesis. Cytokinesis. Okay? Now let us learn. I start with a cell something like this. I start with a cell something like this. This is preferably a young cell. I'm drawing a little bit bigger because I have to draw the cell inclusions. So this is a young cell to understand, and I'm starting with the first phase, interphase. What is interphase? Something like this, see here. A cell which is very, very new. A cell which is young. Newly formed. So एक newly formed cell में, we always start from here. Interphase से हम start करते हैं. A cell which is newly formed. Now, in a newly formed cell, what will be the characteristics? Let us see. The characteristics that we are going to learn, or the parameters that we are going to learn, would be some. And you keep checking on that every time. So, what are the parameters that we have to look into? The parameters that we have to look into would be. The parameters that we have to look into would be number one, the nuclear membrane. These are the parameters we are going to look into. Number one, nuclear membrane. What number two? Number two will be, we'll have to look into the nucleus. That is the chromosome. What is the status of the chromosome? Okay. What next? We'll have to talk about the changes in the centrioles that we get to see. We'll have to look into changes of the cell membrane that we get to see. We'll have to look into the basic chromosome movement. So these are some of the parameters that we'll have to remember when you study the stages of cell. Let us get started with one. Interface. I talk about interface. Now, in case of interface, this is a cell, and okay, if I have to draw the nuclear region, the nuclear region is almost there formed. But what about the hereditary material? The hereditary material, that is the chromosomes, or something like this. Can we count them? No, we cannot, because they are very thin, and now they have to be termed as chromatin threads. Chromatin. So in a young cell which is getting ready to undergo the process of growth, the chromatin threads are thin, extensively coiled, extensively wound up, something like this, always to remember. Okay. What about the nucleus? Is the nucleolus there? Yes, the nucleolus is there. What about the nuclear membrane? The nuclear membrane is more or less intact. The nuclear membrane is more or less intact. What about the centriole? What is centriole? What is centriole? You remember? Centriole. Cell organelle. So ये cell organelle का एक special property याद है? Helps in the process of cell division. The centriole centrosome is a cell organelle which helps in the process of cell division. Is it present both in the animal and the plant cell? Only animal cells. This thing you have learned. So now when we talk about the centriole centrosome का भी status हर stage में याद रखना होगा. So let us see that in case of the interface stage, the centrioles are almost at one side of the cell. Abhi, this particular region of the cell is generally known as the pole of the cell. The anterior to posterior poles. Poles means the two ends of the cell, vertically opposite. The upper end, one pole, lower end, another pole. The poles. So we generally say that the centrioles are at the poles. What is the meaning of that? At one end. Both the centrioles at one pole. Yes, you understand both the poles. But what is a centriole made up of? Made up of protein it is. Centrioles are nothing but proteins. It's a type of a protein known as tubulin protein. It's a type of a protein. So first of all, I talk about the interface. But other than that, what do we understand? Yes, the cytoplasm is still developing to make all its cell organelles. The cell is not mature enough. So now we move on to the next phase, prophase. वैसे तो if I talk about the interface goes through various stages, like there is early interface, mid interface, late interface. मतलब हर एक स्टेज को हम 3-4 सेक्शंस में हम स्टडी कर सकते हैं, because this interface is not going to last only for a few seconds, or it's not a very short period. It's going to last for a few, a short duration, depending on which organism you are talking about. If I talk about our cells, yes, for our cell, it will last for few seconds, few minutes. So when we are talking about, is it just a one flash stage? No, it is a progressive stage. So थोड़ा change, जैसे like the phases of the moon. No, phases of the moon. Does it flash from one crystal state to two crystal state? एक flash में होता है? No, it's gradual. No, same like that here also. It is a gradual thing. What I am telling you is that अब अगर if I am saying that ये जो interface stage है, from the interface stage, when I start drawing the diagram for the prophase, it's not actually a jump from this to this, but it is a gradual stage, meaning I could have drawn in between stages also. And there are in between stages. We are not drawing it because we don't need for the syllabus, but actually here. So now what is about the transitional changes kya ho raha hai when I have to talk about interphase and prophase? First thing is the nuclear membrane has started breaking down, disintegration. So ये disintegration will not happen abruptly. It's a gradual thing. तो मतलब इसमें और इसमें क्या in between stages है? हाँ बहुत सारे. और भी 2-3 stages होता है इसके बीच. So we are not drawing it right now. So try to understand that. So interphase and prophase में सबसे important stage क्या है? सबसे first difference, the nuclear membrane has started disintegrating. Next क्या है? ये जो chromatin threads है, they have undergone gradually the process of coiling, and the process of coiling has happened. And after the process of coiling, now if I draw, after the process of coiling, the chromosomes have become thick, and you can count them now. The chromosomes have become thick, and you can count them now. For example, 4 chromosomes are there. So ये prophase में, it's not only that the chromatin threads have become condensed. ये जो chromatin thread यहाँ पे इतना coil था, it has undergone the process of condensation. It has undergone the process of condensation. So condensation has become massive. So as I said, ये stages होगा. If I show you this way, that ये condensation पहले इतना था, then the condensation became even more. So stage by stage condensation has increased, and now this is looking this way. What else? What about the nucleolus in case of the prophase? It starts disappearing. It starts disappearing. Again, prophase also has got its sub stages: early prophase, mid prophase, late prophase. But we are drawing only one. तो सबसे पहले तो ये हो गया. Okay. Chromosomes, apart from becoming thicker, what else? Chromosomes will also start the process of duplication. Already you know now what is meant by DNA, duplication, chromosome division. And remember, do you remember that we have already studied कि एक chromosome अगर ऐसा है, then the process of duplication would be something this way. Now again we apply this here. So how will we draw the duplication of chromosomes? This way. We draw the duplication of chromosome this way, duplication of chromosome this way, duplication of chromosome this way. So in the prophase stage, in the early prophase stages, पहले तो chromosome thick होता है, and धीरे-धीरे prophase stage के अंदर ही duplication भी खत्म हो जाता है. Duplication is also done in the prophase stage itself. Then what else? What about the centrioles? Now let us check. What about the centrioles? Because these are the parameters you have to remember. What are the centrioles? Centrioles have already started their process of duplication and migration. So duplication and migration also. Now if I say it this way, then the centrioles have also started their process of duplication and migration. This centriole in the later interphase stage, पहले तो ऐसा रहेगा only one. Then they will undergo the process of duplication and then migration also at the other end of the cell. आ रहा है समझ में? So by the end of prophase, what is the structure of the cell like? The cell is almost spherical. Nuclear membrane has started getting disintegrated almost, and the centrioles will almost try settling down at the two ends. Chromosome thickening, condensation is over. Then chromosome duplication is also over. And at the end of prophase, the centrioles are almost this way. End of prophase. Next is metaphase. अब metaphase में क्या होना है? The cell will progressively change its shape. The cell will progressively start becoming oblong. The cell is no more just spherical. The cell will start becoming oblong. अच्छा, इसके अलावा, if I have to talk about some other features, I should say that internally the number of mitochondria has increased, the number of the enzymes have increased, the number of the proteins which are necessary for the process of cell division all have increased in the cytoplasm and the nucleoplasm. These are additional features to remember. अच्छा, अब जब the cell has already entered into the metaphase, nuclear membrane is fully gone. Nuclear membrane is fully gone. And where is the centriole? Almost here. The centriole is almost here, and the centriole gives rise to a ray of protein threads, जिसको ester, these are called esters. Estral rays या तो esters. On both the ends, yes, on both the ends inside is the protein, and these are the estral rays, esters. Getting? Following, no? So now, when we talk about the esters, okay, this way, what else? What are some of the changes? So I draw the esters this way, and now, what about the nuclear membrane? Fully gone it is. And from one pole to the other, this type of tubular network, protein network has been formed. Some tubular protein जिसको हम tubulin बोलते हैं, this type of protein network, microtubular protein, these are thread-like protein chains start getting formed from one pole to the other. From here polymerization happening and touching this, from here polymerization happening and touching this. So ultimately just like a spider का cobweb होता है ना, almost like that. Fine threads of protein start getting building up between the two poles. Then what about these chromosomes? Now, when in the prophase stage, they were just randomly scattered in the nucleoplasm. Now they start getting arranged. Dhire dhire they start getting arranged. So this is what I was showing you about the prophase. Now, if I show you characteristics of metaphase, that's the next stage that I'm talking about. Now, metaphase में क्या होगा? Yes, initially some chromosomes will be here, some hydraulic. So, some chromosomes will be here, and then some chromosome will be here, some chromosome will come this way, some chromosome will be this way. So, dhire dhire they are all getting organized. And in the mid metaphase stage, I said, for each of the stages, there is an early stage, a mid stage, and a late stage. So, हर एक stage में there is something known as early prophase, a mid prophase, and a late prophase. Metaphase में भी early metaphase, mid metaphase, late metaphase. But we are talking about the prominent characteristics only. I repeat again. तो ये तो लगभग जैसे early metaphase में the chromosome pairs, duplicated chromosomes, they are trying to get synchronized, arranged हो रहा है. And in the mid metaphase, what will happen is, in the mid metaphase, all these chromosomes they will arrange like this. In the mid metaphase, all these chromosomes they will arrange where? In the center of the cell known as the equatorial plate or equator, the central region of the cell. They will all arrange like this, where every single chromosome that they have, the chromosome pair that they have, will remain connected by this. Will remain connected to this protein. Something like if I show you. In detail how exactly it happens. वैसे तो that spindle or that network of fine thread-like structure that develops, I already showed you. Now here what happens? Some of this thread will remain connected to this. Some will remain connected to this. Some will remain connected to this. Some will remain connected to this. Actually, it is like this. So we at random keep on drawing it this way. But you have to understand that whenever we are talking about a chromosome pair, I am talking about a chromosome pair. And here the primary constriction of the chromosome. What is the primary constriction of the chromosome? Centromere. So a chromosome pair, if I talk about any chromosome pair, you will understand कि एक जो पोल है वहां से the microtubular thread spindle fiber should be attached to the centromere. तो हर एक chromosome pair will be attached to spindle fibers either of this side or of that side. From here, some spindle fiber will get attached to centromeres of some chromosome pairs. And from this side, the spindle fiber will remain attached to the centromeres of some. For example, say I am saying that from this side, 2 chromosomes are attached, and from this side, another 2 chromosomes are attached like this. जो मैंने पहले डायग्राम ड्रॉ किया था. So it is, if I say that ये एक pair है, then what about the other pair? The other pair will be something this way, where the other pair is this way, and if this is the centromere, and if this is the centrosome, then it will remain attached this way. Are you following what I am saying? Yes. Whatever number of chromosome pairs are there, now that may be 4, that may be 20, that may be 40 for us. When I talk about a cell that we have in our body, how many pairs of chromosomes do we have? 46 pairs, that's quite a lot. So 23 pairs, 46. So now in this 23 pairs of chromosomes, what is going to happen for these 23 pairs of chromosomes? Some of these chromosomes will remain attached to this pole, and some of the chromosomes will remain attached to this pole. Now from each of these poles, what is going to happen? A process known as depolymerization. What is the meaning of that? ये जो spindle fiber, when it was getting formed, what was happening? The spindle fiber, when it was getting formed, simply there were some proteins getting arranged and attached and attached and attached and getting attached this way. So the protein chain was getting formed this way. Now, when the cell has already completed the process of metaphase, now from this end, the protein chain is getting shorter and shorter and shorter by the depolymerization of the molecules of proteins. और ये जो protein chain है, which is the spindle fiber that we talk about, ये इस तरफ से pulled हो रहा है. Follow me? So slowly, if I say that ये जो protein chain that's getting shorter from the pole and it is getting now pulled this way, so what will happen? Ultimately, the chromosome is getting pulled this way. Yes or no? And ये जो protein chain, I said it is getting shorter from this end, and this chromosome pair will be moved from the equatorial plate and it is moving towards this pole. Same will be from here also. Same will be here also. So कोई जो एक chromosome is getting attached to this and it is getting shorter from here and the chromosome is being pulled from here. So there will be one chromosome pair which will be pulled this side. Like this, these chromosomes will disappear from the equatorial plate. They will no more be there in the equatorial plate and they have started moving towards the opposite poles. So, एक pair this way and another pair will be this way coming towards. So, I may draw that these are the two spindle fibers which remain attached to the chromosomes this way. This is how the chromosomes which actually at one time remain attached on the metaphasic plate, how do they start moving? Only by the shortening of the spindle fiber from the pole. Shortening of the spindle fiber from the pole, a process known as depolymerization. होते. Now this chromosome pairs will start moving. But before this chromosome pairs, they start moving. Actually, do they move in total pairs? No, they are not moving in pairs because the pull is from both the ends. The pull will be from both the ends. So this chromosome will start. I'll show you only with one chromosome so you understand. So now I am talking about a pair of chromosomes. I talked about a pair of chromosomes. Every chromosome pair will remain attached to the pole by the way of a spindle fiber. Now, let us talk about a single pair. What happens to that single pair is, to this single pair, there will be the thread from this pole and there will be the thread from this pole. What will happen now? This thread will be now undergoing depolymerization. From both the ends, the thread is becoming shorter and the pull is this way and the pull is this way. So, for every pair, yes, it is getting pulled, but is it actually moving in pairs? No, it's not moving in pairs. We are talking about mitosis. It's not moving in pairs. but this chromosome will split from here. The chromosome will split. This is the chromosome. This is the primary constriction. There will be a slit that takes place here. Then what? Then this chromosome, as they move, one part will go this way and the other part will come this way. This is what is called centromeric division, splitting of centromere, and this starts at this age, anaphase. So progressively what has happened? We had come across that from prophase, slowly the paired condition of the chromosome starts arranging them in the metaphasic plate. Is this complete metaphase? No, this is still not complete metaphase. At a stage of metaphase, what will happen is all the chromosomes, they will more or less arrange themselves this way. This is complete metaphase. But this is only for a short period of time when the chromosomes will start getting pulled from both the ends by the process of depolymerization at the poles. And from every pair, one chromosome will move to one pole and the other chromosome will move to the other pole. Because spindle will remain attached only to the centromere. Does the spindle remain attached to the arms? No. If the spindle would have remained attached to the arms, the chromosome would have got broken. They don't break. Because the splitting does not happen at the arms, the splitting is happening at the centromere. So what are some of the changes we have come across from the metaphase to the anaphase? When we talk about metaphase, yes, the cell is spherical, nuclear membrane not there, the spindle basket is fully formed, the spindle threads arising from the centriole touching the centromere, and by the time now the cell is getting mature to enter into the process of anaphase, this is what is happening. Depolymerization हो रहा है यहाँ पे, and then from every pair centromere divide हो रहा है, centromere split हो रहा है, and the pairs are getting pulled and separated. तो ये जैसे शुरू हो गया, this is what is anaphase. अब ये शुरू होने के साथ साथ the membrane of the cell, now the membrane of the cell will not remain this way. The membrane of the cell will show a furrow this way, and this is a characteristic of animal cells. Furrow formation, जो actually anaphase के distinctive feature है, but वो late metaphase में शुरू हो जाता है. In the late metaphase, it gets started, and it is a distinctive feature of anaphase. So by the time anaphase has started, early anaphase, where the chromosomes have just split, and now they are moving towards the poles, their respective poles. So similarly, after some time, we are going to come across, yes, one and other chromosomes, they have started moving. This and the other, they'll be quite closer to the poles now. And what about the spindle fiber? Are they all gone? No, the remnants are still there. The fibers are still there. Not fully over. So in the late anaphase, what will happen in the late anaphase? This is becoming even deeper. This is a gradual, deeper. And deeper hote when the cell membrane starts almost getting formed, now we enter into the next stage, telophase. We enter into the next stage, telophase it is. See, first of all, this furrow will form and it will deepen, and after that what? We talk about a characteristic telophase. So by the time the membrane has started forming a deeper groove, these spindles are almost disappearing. The cytoplasm is also getting pinched. By that time, the nuclear membrane is again reforming. These are characteristics of telophase. So by that time, the nuclear membrane is again reforming. So what are the changes, transitional changes would be? What about the centriole? Yes, it will be there. What about the nuclear membrane? Yes, it is forming. What about the conditions of the chromosomes? Yes, they are there. Condensed or chromatin threads? No, they are still condensed first. They are still condensed. What about the rest of the spindle? They are still there. They are getting degenerated. And after some time, you will come across, after some time, if I talk about only one part, after some time here, this all will be gone. And at the later telophase, we will see that this same chromosome will start undergoing uncoiling. This will same undergo the process of uncoiling now. It will no more be condensed and thick, but it will start opening up. It will start opening up at the end of telophase, and last is the process of cytoplasmic division when this is complete. When this is complete. So ये pinch होते 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 छोटा होके and will separate. Cytokinesis. Cytokinesis. So after cytokinesis, what is the stage? After cytokinesis, if I am talking about, this is the new cell. For this new cell, what about the nuclear membrane? Yes, now the nuclear membrane is there, fully formed. What about the chromatin inside? All like this. Nucleolus, yes, it is there. What about the centriole? At one end only duplication hua? No. The cell is almost like this cell, no? And very soon this will start the process of duplication. So same will be another cell this way also. Another cell also will be this way where we come across the nuclear membrane and then the deleted material all like this and then the nucleus. So two cells get formed. So progressively the cell starts changing its shape and internal characteristics. So pehle we had studied from interphase to telophase a lot of nuclear changes. No? Nuclear changes have happened. What are we going to talk about these nuclear changes as? Karyokinesis. Karyokinesis. And जैसे ही nuclear changes are all over, now we talk about the process of cytoplasmic division. Cytoplasmic division. What is cytoplasmic division called? Cytokinesis. So what are the two main phases of cell division? Karyokinesis and cytokinesis. Karyokinesis and cytokinesis. Take out your pencils and you will have to draw these diagrams. You will have to draw these diagrams. Okay. Okay. सबसे पहले look into this structure. Try answering this question when I am asking you to draw the diagram sequentially. Take this question, देखो if you are able to solve from whatever you have understood now. Diagram देखो. This type of typical question you will get in your paper. देखो diagram and try to whatever you have understood now. Look into the question and try answering. Go ahead. No, that's what I have been repeatedly telling you. In your syllabus, you are not supposed to write down early, mid, late, kuch bhi. Only one word you are supposed to write. Anaphase have to anaphase. Prophase have to prophase. You are not supposed to write early, mid, late, kuch bhi. Okay? Are you able to identify this stage? So I'll give you first 2 minutes. Let me see how many of you are able to understand. करो फटाफट. Are you able to read? दिखाई दे रहा है? Name the stage of cell division. Name the stage of cell division. Name the stage. Anaphase. These are typical questions you have to get prepared with. You have to understand another type of a question which you very often get. I will ask you now to do. Then drawing the sequential stages. If there's no time today, we'll do it in the next class. Or a sequence. No, I don't want to give you as homework because all of you will do it differently. Drawing the sequential stages, you will do it in the class only. Don't do it in your notebook and bring the next day. Drawing of the stages, I will ask you to do it in front of me. There's a proper way how you should draw the diagrams. It looks very simple, but then often half of the mark is gone. If the diagram is for 3 marks, students end up getting only 1.5. So you should know what exactly to draw, how exactly to draw. So name the parts label 1, 2, and 3. 1 क्या है? Ester. 2 क्या है? 3 chromatid. PID. Chromosome नहीं लिखना. Chromatid. What is chromatid? Arms. My arms. Arms of chromosome. Answer complete. Arms of chromosome. Write one distinguishing feature of this stage. Can you try? Chromosomes are pulled towards poles. And also the cell furrow formation is there. Furrow formation is there. But सबसे distinguishing यही है. Chromosomes are pulled towards poles. Chromosomes are pulled towards opposite poles. Chromosomes are pulled towards opposite poles. Two distinguishing features पूछा है. Then you can write furrow formation B. That's also a distinguishing feature of the Anaphase stages. Okay, let us go to the next. Which is the succeeding stage? Telophase. Draw and label the stage. Chalo, fatafat. Today we finish with this diagram. All of you should be able to do this. Telophase karo, and then next class we have to finish with all these diagrams. Diagram will take a little bit time, so I'm not starting it now. At least 10-15 minutes it will take to complete all the stages to draw. You should be very well acquainted with drawing the diagrams of mitosis, and that comes in the paper. But after I want to give you one more question. Complete first. Draw the stage of telophase and label. अब label कौन-कौन से parts करना है? Esters है कि नहीं? These are the parameters you have to check. Esters है कि नहीं? Centromeres है कि नहीं? That you have to write. That you have to show. Nuclear membrane है कि नहीं? That you have to show. What is the status of the chromosome? Is it in the form of a chromosome? Is it in the form of chromatin? वो दिखाना है. And cell membrane, is it there or not? So these 4 things should be shown in the figure. चलो fast. ये डायग्राम ड्रॉ नहीं करना है. We got the next stage. चलो fast. I want all of you to draw this diagram. I want to see what diagram you all are drawing. Diagram should be neat. In a ruled notebook, get an estimate. Your diagram should be almost 4 lines. एकदम छोटा सा नहीं ड्रॉ करना है. Appropriate size ड्रॉ करना है. Diagram should be bigger. 4-5 lines का diagram होना चाहिए. Diagram should be at least in a ruled notebook. एक 4-5 line का diagram. 4-5 lines मतलब almost this big. ठीक है ना? 1, 2, 3, 4, 5 this big. 4-5 lines का diagram. So that should be an estimate that you always remember. Because most of the students draw this type of a diagram. अरे ये तो 2 lines का हो गया ना? 4-5 lines का. That should be the estimate. Not too big, not too small. Telophase, this is the stage that you have to draw. Telophase में cells should not be saturated. Cytokinesis अभी तक हुआ नहीं है. You can draw the diagram in your array बाप Bio के diagrams, freehand diagrams, the compass of protector bio में use नहीं होता. Because why? Because the cells of our body are irregular. They don't have that sharp edges, sharp sides and edges. Edges नहीं होता हमारे body cells में. You can draw this. You can draw the diagram in your book also. Anyone. You can draw this. You can draw the diagram in your book. Done. Practice code. Diagrams, the shape, the symmetry you should practice. Freehand diagrams you should draw. Not necessary that you have to draw the circle with the compass. Exactly circular हमारे body cells नहीं है, so you should not draw that way. Draw freehand diagrams, but that should be appropriate also. Okay. So you know this diagram for telophase. So what are the characteristics you should check in your diagram? Number one, nuclear membrane should be drawn in fragments. Nuclear membrane is still not complete. The chromosome part, how will you draw? Coiled or uncoiled? Uncoiled. Nucleolus should you draw? Yes. Nucleolus should be there. Nucleolus can be there in telophase, not a problem. In your book, you may come across in the early stages, nucleolus may not be there. That's also true. Do not get confused. Achha, or here, centrioles should be there at one end only. Centrioles only at one end. Should they remain connected? Should the two parts of the cell remain connected by the cytoplasm? Yes. We are drawing telophase. The two parts of the cell should remain connected by cytoplasm. Good. Telophase. So this is the diagram that you are drawing here is actually the late telophase diagram. You have a telophase diagram in your book that is the only your mid telophase utensils. Mid telophase, that the chromosomes have already settled, but the process of cytoplasmic constriction has not yet happened too much. That is the diagram that you have in your book. Either of the diagram you can draw, both are correct. Now, take one question for homework. Are you able to draw? Good. But in the light view, the diagram should be prominent. See, your nuclear region is not prominent. Nuclear region for a lot, अच्छी तरह से करो. The nuclear region should be almost in the center. Why it is like this? When you wait a minute. See the nuclear region, it will be almost in the center this way, almost in the center this way. Nuclear region fragmented and the chromatin threads inside. Huh? So that correction to it nicely. Diagram should be drawn very neatly and nicely. Nothing but question. Draw the metaphase of mitosis in an animal cell. Draw the metaphase stage of mitosis. Draw the metaphase stage of mitosis in an animal cell with 4 chromosomes. With 4 chromosomes. Now, we will draw the diagram very neatly with all the parts labeled. But 4 chromosomes, what do you understand? Do you understand the question? Draw the metaphase state. That you understand metaphase state draw करना है. तो जैसे ही मैंने metaphase बोला cell का shape, you should understand कि cell का shape मुझे कैसे draw करना है. What should be the cell shape? नहीं, circular नहीं. नहीं, metaphase में कहाँ से furrow रहता है? In metaphase, the cell shape should be something this way or something this way preferably. Oval थोड़ा सा. Spindle रहना चाहिए. Nuclear membrane नहीं रहेगा. Nucleolus नहीं रहेगा. Esters, both the poles में, opposite poles में. And what will you do with the chromosome number? That is what I want to know from you. 4 chromosomes मैंने बोला है. तो क्या draw करना है? 2 pairs की 4 pairs? 2 pairs. 4 chromosomes, 4 pairs. 4 pairs. Because we start with the parental chromosome with a number 4 chromosomes. तो वो in the later process duplicate होके 4 pairs, 4 chromosome becomes 8. So when I give you 4 chromosomes, you have to understand it is 4 pairs 8. 6 chromosomes, 6 pairs 12. 3 chromosomes, 3 pairs 6. This is what you have to remember. So these are the 2 difficult type of questions which you get in your paper. ये करके now this remains as a homework. Complete this at home. A neat diagram you draw. The next class when we meet again, all of you should have your pencils and you again draw the sequential stages of mitosis. That's very important. Meiosis stages not needed, but only some differences we have to study. We wind up here.